Now, if you've been here before, you'll know that the Green Air Hub project officially started in September 2022 as a coordination and support action of the European Commission under the Horizon Europe programme. And it currently represents 15 EU ERA nets, co-funds and self-sustained initiatives in the field of agri-food and biotechnology. We started these breakfast clubs here last year in October, and we've had 4 very successful series so far. The first was Circularity in Agriculture. The second series was related to biotechnology to advance sustainable solutions for bioeconomy. The third was Sustainable Food Systems, and the one that we finished at the end of 2024 was Precision Agriculture Technologies for a Sustainable Future. If you're interested in listening to the talks related to any of the topics from these series just mentioned, please do check out our YouTube channel where all of the videos are available. Now, for the past 3 months, we've been focusing on the topic of plant health and protection, where we've hosted a series of expert-led discussions aimed at fostering a deeper understanding of plant pathology, integrated pest management, and sustainable agricultural practices. Just a few housekeeping announcements. Please feel free to type your questions in the Q&A chat section, which I've enabled for everyone. I'm going to do my best to address as many questions as possible within the time that we have this morning, which is 1 hour. If at the end of the session you have a question for either of our speakers today, please do remember to address the speaker directly in the question. And we'll open our session this morning with Katharina Heinrich and Roy MacArthur from the Food and Environment Research Agency in the UK. A senior scientist at SIRA, Katharina Heinrich specializes in stable isotope analysis across diverse matrices from agricultural produce to insects. As part of SIRA's food authenticity and profiling team, she leads project management and overseeing the delivery of analytical services, including those related to CBD products and insect bioconversion. She's contributed to multiple European research initiatives and collaborative research projects, and has authored or co-authored over 15 peer-reviewed publications in her field. Katharina's co-presenter this morning is Roy MacArthur, a statistician at the Food and Environment Research Agency, also with a strong background in analytical chemistry, plant biosecurity, and food safety. Roy has over 1,800 citations, an H index of 26, and has co-authored numerous influential works on topics ranging from pesticide residues to virus detection in crops. He collaborates widely across academia and government agencies, contributing to the advancement of food and environmental research. Today, Katharina and Roy's joint talk is entitled Use of Stable Isotope Ratio Analysis for the Identification of Invasive Species Native in Alien Environments, which is part of the UFresco Phytosanitary Research Initiative. Katharina, Roy, thank you very much for accepting our invitation for a webinar series. We're absolutely delighted to have you both join us this morning. Without further ado, I'm going to stop sharing my screen now so you can start sharing your presentation. Thank you very much.
Thank you much, Barry. Lovely to see you all or meeting you all and giving the opportunity to present our project we had in the last 5 years. So I'll share the screen now. With our presentation. So as I said, we talk about stable isotope ratio analysis for the identification of invasive species native in alien environments. It was a collaborative project with colleagues from Austria, Italy, the US, and France. And it ran between 2018 and 2023. And with Infera was a big collaboration because we have, as you mentioned, you've got our statistician Roy with us and Larissa Collins, who is a lead entomologist, and then myself as the analytical chemist doing the technical measurements. And now. Brilliant. On to the next page. So just to give you a bit of a context about our success story, where we used the forensic technique to differentiate origins of invasive beetles, carry on with what if scenarios, why we want to use our technique, our overall project goal, Then move on to the sampling location where we got our beetles from, what stable isotope ratio analysis actually is. And then I pass on to Roy talking about our results and the final conclusions of that project. So Asian longhorn beetles, our example species, is an invasive wood-boring insect. And what are consequences if this invasive species is present in Europe? So one of the main issues is that we've got tree loss, biodiversity loss, if we're moving into environments of our native insects. This causes then ecosystem disruptions, influence economic and aesthetic costs. And that brings us, what are then the costs for taking action against those invasive species? As I said, this economic cost, aesthetic costs, if you have to fell tons of trees around the area to extinguish this invasive species. And obviously you lose your trees by doing all this felling. But what is when you actually had a test to differentiate if that beetle is a tourist, just arrived, or is already resident and is populating the various cycles of his life? So one option is don't take any actions. If the test says it is not a resident. The other option would be take action only if the test says it might be a resident. And why I want to ask that question is because we want to maintain protection by taking actions when it is needed, so to save costs when we don't need to take any actions. And our main project goal was to make a test that actually works. So on the top, you see a little isotopic mannequin that tells you that everything person's eating, any animals, the signature of the food they're taking was represented in the isotopic signature. So it's from carbon, oxygen, nitrogen, or hydrogen. This is the reason why we want to develop stable isotope ratio analysis as an arthropod tree pest determination tool. This is used to obtain reliable information about the origin of exotic pest species. And this helps us the ability to make this determination as part of the decision-making process. So government is very much interested how we can solve these issues and saving costs. And this moves on to actions and measures to be taken in case of interceptions or outbreaks of non-native species. So that's the preamble why you undertaking that work. So we've got a lot of project, but you also need to get some samples. So for the Asian longhorn beetle, this is in the name. So it's native in Asia, and specifically a French student went out doing her PhD to China to 11 locations and sampled adult beetles living around there in 2016. And then we compared those samples against Asian longhorn beetles found in outbreaks in Italy from 6 different locations that was being sampled in 2019. So we got our two populations and what's the best way to analyze these to determine geographical origin. We're using stable isotope ratio analysis. That's a very established technique for food authentication, geographical origin in foods, for example, beef or wines or fruit juices or honeys. And how we use the data and how it's generated is, for example, we presented here on the top left corner is the hydrogen, one of stable isotopes. The other stable isotope available for it is the deuterium, and the only difference is the presence of additional neutron in the nucleus. And we're actually looking at the ratio of the heavy isotope against the lighter isotope. And this makes it interesting because obviously hydrogen 1 has got a presence of 98% in the environment, and the deuterium is only 1.something%. So we got, if you look at the ratios of the heavier to the lighter, you got tiny little numbers. And you compare these of the ratios we find in the sample against the theoretical standard with their ratios. And because the numbers are so tiny, we multiply by 1,000 and express that as a per mil delta value. And in the case for hydrogen, the set delta value set to 0 is the Vienna standard mean ocean water. So that's our preamble for what delta values are in stable isotope ratios. So in case our samples will have less deuterium present, they will get a negative value. If the samples would have more deuterium present than the standard mean ocean water, you get a positive value. So it's just to give you an idea what numbers will look like when we look at later on the results Roy is discussing. So, and then moving on, what's the instrument schematic looks like. So on the bottom, we can see there, so I said before, we don't need much sample material. So 1 milligram is sufficient. That's tiny amounts. So we weigh them in little tinfoil capsules. They're crumbled together, so even smaller than like black peppercorns or sesame seeds. They're really tiny. They get dropped then into the instrument, which holds a high temperature furnace at 1450 degrees. So it's glowing hot, an excess presence of carbon, and everything gets turned into carbon monoxide and hydrogen. This goes through the elemental analyzer into the different gas traps, and they get split up via the diluter into the ionization for the mass spec. In the separate gases, we have then hydrogen and carbon monoxide. And on the bottom, we see in the schematic here the chromatogram for monitoring gases of the hydrogen as a reference gas to check what we are measuring. And then on the bottom here, we got the sample peak. So it's a nice clean line because there's nothing else. There's only hydrogen or carbon monoxide going into our system. So this is a brief nutshell how the stable isotope ratio mass spectrometer works. And that brings us to how we can use that to find or determine geographic origin of any living creature. So the University of Utah are very big in generating isoscapes. For example, here's a precipitation map of hydrogen on the global scale. And as talked, our theoretical standard is the ocean mean standard water. That's around the equator. So we got the set to 0 around in East Africa. And then we're moving on to more depleted areas from the orange to the yellow to the green and to the blue. And then we look at our own samples we analyzed. We've got Italy and China, so in the yellow area. And that leads us to our China's beetles. We measure between -77 and -116 per mil, while the European beetle in Italy was between -61 and -90 per mil. So there's a difference, but there's also an overlap. And to make it more meaningful evaluation, we bring out our statistician, and I move on now to Roy. So I'll just put on the next slide and you say when to move on. Thank you very much so far.
Yeah, so hello. As Katrina opened, the question that we asked was, well, what if we had a test? And that's probably the first question to ask, when you're thinking about developing a test, when you're thinking about should we apply a technological or a technology to a particular problem, then the first question to ask was, well, if we did and we had a test, what could we do with it, and what would its performance have to be like in order for it to be useful, and how quickly would it have to work, and all those kinds of things. And so what we're doing here is we're looking at how we could use these measurements, these stable isotope measurements of hydrogen, in a test to determine whether a beetle is a resident, which is bad, or a tourist, which might be okay. And because being a resident is bad, we want the result, we want the test to always tell us if something really is a resident. We want the test to always raise the alarm and say, this might be a resident. If it's a tourist, then, well, it would be nice to know it's a tourist, because then that could save us money and we wouldn't have to take action. But it's kind of less important to always be right in that case. And so that's another part when we're designing a test, is to be clear about which way is more important and what we always want to be right about. And in this case, we really want to be right if we test a resident, we absolutely want to know it's a resident. We care a lot less about the other thing. So what we did is we took all that into account, and we took the results of European beetles, residents, and Chinese beetles, who would be tourists in our scenario, and they were clustered in different locations, so they weren't just a simple random sample from across the two populations. And we took that into account in a statistical analysis and estimated the statistical parameters, the mean and the variation for each of the populations, for if we had taken an individual at random from across the population. And so to do that, you have to take into account things like the degree of clustering, the within and between clusteral variation, and then you make an adjustment for the what that does to the effective number of samples you have in degrees of freedom and things like that. So fine, we can do that, and then we end up with two distributions: a distribution for the resident beetles, which is the more negative one, and then a distribution for the tourist beetles. And so those distributions, our best estimate of those distributions, are represented on these figures by the lines. And then you'll notice that actually there's quite a wide kind of fuzzy bit around the lines, and that is the uncertainty about what the actual underlying distribution is. That comes from the fact that we tested a few individuals from each population rather than the whole population. So fine. So we've estimated two things. We've estimated where the edges of the distribution are, and that's the line, and then we've got an uncertainty about that as well. And so those are the two things we need in order to work out, well, I suppose, what we think the performance of the test is and what the performance of the test might actually be, and probably also what might be the value of getting some more information and making those the uncertainty less. So we want the test to be very reliable for detecting residents, and so traditionally that means at least only a 5% chance of thinking that a resident is a tourist. And so the first thing we did was set that threshold at 5%, and that's what's happening in the top left figure. That's the probability of getting the wrong answer is set at 5%. And then we set our threshold measurement result based not on where the central line, the central estimate was at 5%, but at where the kind of the 95% confidence interval for what the probability is less than 5%. So we're taking not only our best estimate of what's going on, but the uncertainty in it as well, to make what is quite a conservative test that says it's not we're going to be wrong 5% of the time. We're definitely going to be wrong less than 5% of the time with 95% confidence. So it's probably a lot less than 5% wrong. And that's how we're going to set our threshold. And then we compare that threshold to our tourist distribution, and that tells us, again, taking the uncertainty into account, that we will correctly decide that something is a tourist, maybe up to 50% of the time, but it could be a bit less, quite a lot less actually. So we'll be right about 20 to 50% of the time, and we'll say, no, this is a tourist, we don't need to take action. So that's how we analyze the tests, the results that we've got to say, if we built this into a test, what would the performance be? And then we can use these probabilities that we've estimated, the probability of getting it right when it's a tourist, the probability of getting it right when it's not a tourist, in order to estimate what the costs and benefits are of applying the test, which might be on the next slide. Yes, it kind of is. So here are some options for how we can use the test. So two kind of important options. We can decide not to bother. We just don't use the test at all. And in that case, we've got two options. So one is we always assume everything's a tourist. And the other option is we always assume everything's a resident and we need to take action. So that's a very cheap option from a test point of view, because the cost of doing that is nothing. It means that under one of those options, we always have to take action. So we're maxing out the cost of taking action. But in that scenario, we don't lose, we never get the kind of cost of not taking an action, which is an outbreak. Or we could not use a test and always assume that everything's just a tourist and we don't need to take any action. So hooray, no cost of testing, no cost of action, about the full-on maximum no protection cost of an outbreak. And then we can do two other ways of using the test. We can actually use the test and say, well, I don't like any of those. I don't like maxing out those two costs. Let's try and use a test. And then we got the other two scenarios. We can use the test, assume that it's a tourist unless the test says it's a resident, or the other way around, assume it's a resident unless the test says it's a tourist. And so because we've worked out all those probabilities and we've worked out how the test is going to perform, we can then work out what proportion of times we're going to be right and wrong under those scenarios, and what the costs are likely to be, as expressed as a kind of proportion of the 100% costs of not using a test at all. And so that is how we did. That's how we've presented the performance of the test, so that people whose job it is to make a decision about whether it's a good idea to apply this test and whether it's worthwhile are able. We've given them the full information, so they're able to make that decision. Should we use this test or not? Is it useful? How much will it cost us? And further, because we've included the uncertainty in what our estimates are, we can also get a good handle on, okay, this is kind of looking maybe promising, but we need to do a bit more work to reduce that uncertainty. Then we've got a handle on that, what the potential cost improvement will be by reducing the thickness of that uncertainty, because part of the threshold is because we've got uncertainty, and so if we reduce the uncertainty, we get a better threshold that has a more favourable kind of cost benefit outcome. So that's what we did, and that's how we presented the performance of the test.
Hey, Katerina, thank you so much for such an interesting and insightful presentation. I just want to remind everyone that the chat box and Q&A section is open. So please do feel free, if you have a question for either Katerina or Roy, to type that question in. Just while we're waiting for questions, Katerina and Roy, just a couple of questions I had of my own just by listening to your really, really interesting presentation. The first question I had relates to kind of environmental factors and how they play out in terms of the type of project that you're doing. And I was wondering specifically what kinds of environments make it either easier or harder to use stable isotope ratio analysis to track invasive species.
Can I start with it? So it makes it easier to track if obviously invasive species comes from a totally different climate. That would help, obviously. I showed you the map with the hydrogen isotopes. Or whatever the species lives more in the mountainous region, so therefore also the values are more depleted compared to someone close to the ocean. So it's easiest way to differentiate is if they are completely different climatic changes in environments. That helps with the differentiation.
And Katerina, how do changes in the environment, like pollution or land use, how do they affect, if at all, isotope data?
Pollution can affect maybe nitrogen values. If you would be looking for those isotopes, they can have an effect. So we just focus on the hydrogen because it just gives a more geographical indication. But if we would know, for example, not these beetles or other species, they're close to any pollutions, and you would look for other stable isotopes for the nitrogen or the sulfur in case it's more sea spray present or something, or volcanic activation was happening in the past. So we can put those in. And then moving on, what we haven't done, other research groups looking also at lead stable isotopes more recently to pick up on the environmental pollution. Or if you got, because insects are quite tiny matrices to work with. So if you have more bigger animals like cattle or chickens or whatever, so you would go in on the pesticides route or environmental contaminants, not using stable isotope, but other analytical techniques to find out where things are coming from.
Very good. Thanks, Katerina. And Roy, you were mentioning just about the estimated cost involved in kind of rolling out this form of analysis on a kind of a large scale. I was just wondering, could you give us maybe an insight into cost? Like how expensive or even time consuming is it to do stable isotope ratio analysis on a large scale?
Okay, so yeah, I look like I gave an estimate of cost, didn't I? But,
maybe that's more a question for somebody else entirely.
no, but I didn't exactly. So what I did is, so they were proportions. So it was like, if you know what the cost of an undetected outbreak is, then this will reduce that cost by 50%. If you know what the cost of having to do action on every outbreak is, then it will reduce that cost by 20%. And then there's the cost of doing the test. So I don't know what the costs are, but I'm able to tell the people who do know what the costs are what the test will do in terms of the proportional reduction in the cost. And so that's how we presented the results. So no, I don't know what the cost of
I was just
dealing with
just being nosy.
Outbreak is. I bet it's huge, though.
Roy,
I could say,
sorry.
So with the outbreaks we had in Kent, I think was in the millions to eradicate all the trees and felling them, kind of thing. Yes. And an IRMS test can be around £250 a sample, kind of thing, in that ballpark figure.
Excellent, thank you. So just a couple of questions coming in from our audience. Quinton asks, when you decided to use isotopes, H isotopes, you made an assumption on the isotope ratio of the beetles. Did you validate your expectations? Did the values you measured in beetles from China and Europe fit to the ISOSCAPE map? A couple of questions there.
Very simply. Didn't actually compare the ISOSCAPE map. We just compared our beetles. We wanted to differentiate between those two different groups.
Excellent, very good. And just another question from Anonymous. What kind of costs are to be expected for these analyses, order of magnitude? Again, I think you probably just touched on
I just mentioned that,
there, that it was just, impossible at this stage to say. Katerina, Roy, thank you so much for your, first of all, for just such an interesting and excellent presentation. My God, really, really interesting, excellent science. So now joining us, as I say, is Dr. Negin Ibrahimi, a senior research scientist at the Flanders Institute for Agriculture, Fisheries and Food in Belgium, where she specializes in nematology, invasive pest management and phytosanitary research. In particular, her work focuses on developing sustainable control strategies, rapid detection methods, and monitoring systems for quarantine pests and agricultural threats. She has extensive experience in integrated pest management, nematode population dynamics, and stable isotope analysis for tracking invasive species. Dr. Ibrahimi has led and contributed to numerous international research projects addressing challenges in quarantine pest identification, fruit fly detection, and plant disease epidemiology. She has published extensively in scientific journals and actively collaborates on research supporting biosecurity, crop protection, and sustainable agriculture. Today, Dr. Ibrahimi's talk is entitled Fast Detection Methods for Quarantine Tephritidae. Megan, thank you very much for accepting our invitation for our webinar series. Again, we're absolutely delighted to have you join us today. So the floor is yours. I'm going to stop sharing my screen now so you can start sharing your presentation. Thank you very much.
Thank you very much, Barry. Indeed, thank you for inviting me to this webinar. I'm very happy to have this opportunity to present the outcomes of our small IFRESCO project on the fast detection methods for quarantine Tephritidae. And the project acronym was Tephrifade. Well, as I mentioned, it was a small project. It started in June 2022, and it lasted for 2 years. We had a large consortium, 19 partners from 14 countries and across different continents, as you can see here. Well, why this project was proposed, but yeah, you know, these fruit flies belonging to the family Tephritidae are one of the most diverse families of Diptera, with over 5,000 described species. And it was in 2020 that EFSA performed a pest categorization based on which 257 species were considered as potential quarantine pests for the EU, among which we had Anosrepho ludens, Bactrocera dorsalis, Bactrocera zonata, and Ragoletis pomonella in the list of priority pests. And most importantly, we have seen that between 1995 and 2019, there have been more than 4,300 interceptions of these non-EU Tephritidae quarantine species on several crops, fruit and vegetables, and especially the perishable goods like mango and capsicum. And these fruit flies can cause severe damage to fruit and vegetables. That's what I should really emphasize on that. And well, it was just recently that there has been a kind of shift in the EU policy from some, I think 2 years ago. We are required to identify the intercepted Tephritidae at the genus or even species level, unlike before that we could just indicate them as the non-EU Tephritidae. And you best know that the reliable morphological identification is pretty difficult for this group. It's possible for the adults and the later stages of the larvae, but when we are dealing with the most frequently stages intercepted, which are the earliest stages of larvae and sometimes even pupae or eggs, then morphological identification is not possible. Therefore, we certainly need to look for alternative methods that can be accurate and at the same time pretty fast, because as I mentioned, we are dealing with the perishable goods and we cannot keep them long at the point of entry if we have to test them. These methods, well, we thought of the molecular methods. They can be LAMP, loop-mediated isothermal amplification, qPCR, or barcoding. Well, this Tephrifade project aimed to compile and gather all the existing fast diagnostic methods for identification of this group and to facilitate the best practices of these actually fast identification methods and organizing also some interlaboratory tests to validate and harmonize these methods. We also worked to identify the taxonomic gaps in the available DNA sequences, and we really hoped to collect these missing species, the species for which no DNA sequence was available, and provide their sequencing to fill the gaps. Well, in the first phase of the project, as I mentioned, we gathered all the fast diagnostic methods which were published or were being developed during the time that the project was running in different labs. And the one key observation was that these fast molecular identification methods are only available for a small number of Tephritidae species. Here I have a little summary of the methods. As you can see here, for example, the LAMP assay is only available for 12 species, qPCR for 15 species, and multiplex PCR only for 5 species. And in the case of CRISPR-Cas, which was a recently published method, it is only available for 2 species, Bacrocera zonata and Ceratitis capitata. Well, our literature work also showed that barcoding remains the most commonly used method in different labs for identification of Tephritidae. We also decided to review all these published papers and exchange the information and summarize all the information regarding these molecular methods in a little file. And as you can see here, we assessed which stages of the life cycle were used for testing, what DNA extraction method was used, and most importantly, we asked the partners whether any of these methods are being used in their lab, and also how long each of these methods might take. And again, I should mention that we notice, except for a few labs, in many of the labs, in actually most of the labs, barcoding was the only method used for identification. We decided to evaluate some of the LAMP assays for the most relevant species that we are actually intercepting in our lab often. We opted for the LAMP assay. LAMP assay, LAMP, loop-mediated isothermal amplification, is kind of similar to the conventional PCR, with a difference that here the DNA amplification occurs at a constant temperature, and here in a set of 2 primers that we usually use for the conventional PCR, we are using 4 to 6 primers, which makes the method pretty specific and sensitive. And the biggest advantage of this LAMP method is that it can be used on site or in field. Well, the first method that was actually evaluated even before the start of the project, but by our colleague, Dr. Van Estermo, in the framework of another project, Tephrifast, was a LAMP assay for identifying Ceratitis capitata. But unfortunately, we saw that primers were not specific enough. And within our project, we had a recently developed method by Zhang et al., and that was designed to identify different Ceratitis species, Capitata, Cosira, and the FARC complex. We tried this method, and here they used a kind of kit warm start colorimetric lamp, which is making the method very easy and user-friendly if you are supposed to use it, for example, on site or in field. The kit contains a dye, and upon the amplification of DNA, you have the change of color due to the change in the pH of the medium. For example, here you see that we try to evaluate this method. As you can see here, when we use the Ceratitis Cosira primers, and we had Bacrocera dorsalis as a non-target species, as you can see, there is no change of color in the case of dorsalis, Bacrocera dorsalis, while in the case of Ceratitis Cosira, we had a change in color from pink to khaki. And it worked also in the case of Capitata. However, unfortunately, we didn't succeed to reproduce the same results for the Farc complex, Ceratitis coelishi, rose, and the two species that we had from Farc complex. We also evaluated a very simple method, but very user-friendly multiplex PCR assay to identify again Ceratitis species as well as Bactrocera dorsalis. You see the different sets of primers that they had. We need to modify the cycling condition a little bit. And fortunately, as you can see here, in the case of Ceratitis species, we were able to produce the same amplicon size that was mentioned on the paper you can see here. But again, here for Bactrocera dorsalis, we didn't reproduce. We were not able to reproduce the same results as the others. But it was honestly among one of the most promising methods when we did the validation in our lab. Actually, a very good event that happened during our project was a 3-day workshop that was organized by our very partner, MVWA in Wageningen, with many thanks that go to Bart van der Vossenberg and Emil Sleis, who hosted us over there for 3 days. We had participants from our consortium and also outside of our consortium. And during this period, we had MVWA that led a session on high-throughput sequencing and data analysis in the case of Tephritidae. Afterwards, Vanes, our molecular expert, demonstrated the LAMP assay method for Ceratitis cocera that he had developed in the lab. And finally, our other partner, Royal Museum for Central Africa, introduced methods, the SNP analysis for the origin tracing of Tephritidae. And most of these sessions were hands-on, and they were quite well received by the participants. Shortly after the workshop, we also performed a test performance study to validate and actually to evaluate the efficacy of the LAMP assay that Van Estermo had already demonstrated during the workshop. Here I briefly describe how the method works. He opted for an easy, user-friendly extraction of DNA from the larvae. You can see here, first he cuts the anterior part of the larva, then he divides the larva into 2 pieces and transfers them to kind of plant lysis buffer. He vortexes the sample only for 1 minute, and then he incubates the sample at room temperature for 5 minutes. Pretty easy actually, and within a very short period. Then he uses 2 macroliter of the 1/10 dilution of this lysis solution, and then he adds it to the LAMP, the species-specific primers, the LAMP primers that he has designed, and the isothermal master mix that is provided already in the kit. And then to visualize the amplification, you have different options. You can use either the portable device, which is called Genie, with different versions, Genie 1, 2, or 3. And if you don't have that, then you can use a qPCR machine in your lab. It's a very fast and sensitive method, actually, indeed. Well, that was the method that was explained during the workshop. As I mentioned, we ran a TPS. Actually, for this method, we provided the participants. We had 10 participants in this test. We provided them with the LAMP assay protocol and a panel of 6 DNA samples. In the protocol, we outlined the different steps of the process. And fortunately, 8 out of the 10 labs succeeded in the test performance study. And the 2 labs that failed, one of them, it was only due to the wrong synthesis of the primers. And when they tried it with the new batch of the primer, it worked. That was quite happy news for us. In the next phase of the project, we try to recover all the available sequence data for these Tephritidae species. We retrieve them from different databases, both NCBR, GenBank, and also QBall, and all the sources that we could have access to. And the purpose was to create a list of Tephritidae species that lack DNA sequences data, actually. While accessing these databases, we also try to indicate whether we have the complete genome of these species that were listed by EFSA, I mean the 257 species. And also we recorded the number of sequences available for different target genes. And we came up with a relatively long list of species of Tephritidae for which we don't have any sequence data available. You can see here we had 69 species lacking sequence data in GenBank and 80 species in BOLD. That is relatively a high number. Within this project, we had promised that we do our best to locate some of these species at least, but unfortunately, despite our best attempt, we were not able to find the species and provide their sequences. In summary, we saw that while barcoding is the most commonly used method for identification, these fast detection methods are really, they remain very limited to a very small number of Tephritidae species. The evaluation of the published techniques actually validated some of these methods, promising methods, but we also noticed that we certainly need to have a thorough assessment, validation, and verification of these methods before implementing them in the lab. And to do so, we really need a framework for validating and standardizing these methods. The successful TPS test also for the LAMP assay also is kind of triggering us to ask to include this method in the EPO protocols, and we hope that it will be adopted by EPO soon. And we have a list of the lacking sequence species that lack sequence data. And here I can announce that hopefully there might be a continuation of this project, but this time more technical. The main objective of this new project, Tephritidae 2, will be to develop rapid identification methods for more Tephritidae species, especially the ones that are standing on the priority list, create a framework to validate and also to harmonize these molecular diagnostic methods across different labs, and organize more targeted collection efforts in regions where we have this missing species. And we hope that we will have more partners to join us, and hopefully we can move forward to shed more light on these methods. Thank you very much.
Megan, thank you very much for such an interesting and insightful presentation. I just want to remind everyone that the chat box and Q&A section is open, so please do feel free to type a question for Dr. Ibrahimi at any point. Just while we are waiting for one or two questions to come in, I just have a couple of questions of my own. Please forgive them now if they're very basic. I was just kind of really wondering about the transmission of Tephritidae. So how do these fruit flies, how do they typically enter or spread into new regions? I'm presuming it's primarily trade. Would that be correct?
Yeah, it's mainly through the trade of fruit and vegetables. There are mainly the exotic species, and they come, if there are loads or cargoes of fruits like mango, we usually have them there. But we should also keep in mind that we shouldn't underestimate the hand-carrying fruits and vegetables. Sometimes you can have them also in the luggage of the passengers. Yeah, these are the main pathways indeed.
Excellent. Just a couple of questions from our audience. Rakesh asks, what should be the concentration of pests, and what stage can we identify the species on site using LAMP technique?
These LAMP methods are usually, when we validate the methods, we usually we try to use the different life stages. Normally, it should be possible to identify all stages of the life cycle using these LAMP methods. And for concentration, it depends. Yeah, you do a sensitivity test usually to determine the lowest amount of DNA that can be detected using your method. And it's usually depending on the different LAMP assays. Each LAMP assay has its own limit of detection. Yeah.
Mm-hmm. Very good. And just a follow-on question again from Rakesh, how do you collect samples for the on-site tests?
Well, how do we collect? Usually, when I talk of on-site tests, well, it's, for example, at the point of entry, right? We get them at the National Plant Protection and MPPO in Belgium. And in the lab, you can just collect them from the infested plant material, like the mango fruit. You make sure that you avoid contaminating the sample because, as I said, the LAMP assay is very specific. It's very sensitive and specific, but at the same time, it's very prone to cross-contamination. And you just have your larva out of the commodity. If
Excellent.
I understood this question well, of course.
Yes, no, I think that sounds correct. Megan, just a question from Roy actually. He was just wondering if we wanted to make it even faster, what might be the best way to do that?
You mean if there is another method that can be faster than LAMP assays?
I presume yes.
Yeah. Well, faster than LAMP methods. Maybe, I don't know if, for example, I know that recently MinION is high throughput sequencing using MinION nanopore is being discussed a lot to be used, but I know that we need a very strong data references to go for that. And this is a method that our molecular expert, Vanessa Dermo, is going to investigate in another project, RAP-ID, to see the possibility of using MinION for identification, because there also we are dealing with a very portable little device, but I guess it's more expensive than the LAMP assay.
And
but I can,
Sorry, I was just going to say, Roy was just saying, is it sampling that takes the time?
Sampling, normally, if you are taking it out of the plant material, it shouldn't be very time consuming. Of course, it depends on the condition of the plant. But for example, in terms of mango, we usually have quite a large number of larvae when they are infested in the plant material. But sampling can take time depending on the status of the sample, of course. But
Megan,
I know that
sorry.
even it is possible using this LAMP assay. I remember that one has tried a method that even blending your sample to see whether you can extract from the plant material that contains the larvae of the target species. That might speed up the process a bit more, but usually, I guess in terms of sampling from the commodities, it's not a serious problem.
Excellent, very good. And Megan, I suppose just a follow-on question from that. Are there any new technologies that you know of on the horizon for even faster or smarter detection of Tephritidae?
Well, at the moment I can't think. I thought the MinION might be an option. I know there are also some methods which are not based on DNA, like multi-top spectrometry, that you measure the mass of the proteins, and then you create a species-specific protein fingerprinting, and in that way, it might be one of the methods, because it's apparently, if you have the setup, it's pretty cheap and quite fast. But as I mentioned before, for both MinION and this MALDI-TOF, you need to develop a very strong database before starting.
Megan, thank you very much. I think we'll just leave it at that. It's just 2 minutes to 10. Thank you very much to our audience for your questions. I'd just like to thank once again Our three excellent panelists this morning, Katharina Einrich, Roy MacArthur, and Negan Ibrahimi, for giving us really wonderful and insightful presentations and tremendous overviews of the respective projects this morning. So we really appreciate the three of you joining us, and thank you so much. Our next webinar will be on April the 24th, when we'll begin our new webinar series actually on animal health and welfare. I'll be sending out the invitation to all of you in the following days. In that particular breakfast club in 2 weeks' time, we will have Professor Christine Van Rieth from the Faculty of Veterinary Medicine at Ghent University talking about emerging porcine influenza and coronaviruses, and Professor Tom McNeely, Scientific Director of the Morden Research Institute in the UK, discussing the development of a pan-European network to understand how Coachella bruneti genotype determines pathogenicity and zoonotic potential. Again, thank you very much to our panelists this morning, and thank you everyone for attending and for your questions. We really hope you enjoyed the webinar this morning, and we hope to see you again in 2 weeks' time with us. Thank you very much.